Nous allons donc poursuivre notre voyage entre neurofeedback et neurosciences cognitives, en nous intéressant plus particulièrement dans la session qui suit au neurofeedback guidé par IRM fonctionnel. Et pour cela, c'est un immense honneur de recevoir aujourd'hui le professeur David Linden. Le professeur David Linden ne travaille pas, comme c'est indiqué sur le programme, à Cardiff, mais à Maastricht désormais. C'est un des pionniers du domaine, puisqu'il s'est très tôt intéressé aux applications potentielles cliniques du neurofeedback guidé par IRM fonctionnel. Il a coordonné le projet Braintrain notamment. Il a été l'auteur de publications très intéressantes sur le neurofeedback guidé par IRM fonctionnel, notamment dans la prise en charge de la dépression. Voilà, professeur David Linden.
Good morning, everyone. It's an absolute pleasure to be here. Thanks very much for the kind invitation. And you'll forgive me for speaking in English. Although I was able to follow most of the previous talk, which I think set me up very, very nicely, because I was going to talk more about the question, what has or can neurofeedback do for cognitive neurosciences? You heard a lot about how background in cognitive neuroscience is essential for our approaches in neurofeedback, and I think many of the principles that we heard about apply in exactly the same way in fMRI and EEG neurofeedback. And you also heard about what neurofeedback has already done for cognitive neurosciences, although you said that we know much less about that, and I agree with that. But this idea of using neurofeedback as a new tool for understanding of brain behavior relationships, I think, is a very interesting one. And I'm referring to mainly the last part of the previous talk. And I'm going to talk about similar studies that have tried to address this from the fMRI neurofeedback point of view, although I agree with the previous speaker that really this field is just at its very start. So the basic idea in terms of applying neurofeedback as a tool in cognitive neurosciences is, I think, to address the problem of reverse inference. In classical neurofeedback, in classical cognitive neuroscience studies rather, 95% or more of studies that we do and read entail some sort of perceptual or cognitive manipulation and then measure brain signals in a correlative manner. And that is, of course, very, very interesting, but it tells us something about what happens in the brain when we engage in a particular cognitive process, or what can happen in the brain, but it tells us relatively little about the causality of these brain changes. Now, TMS and other neuromodulatory techniques have been introduced already a while ago to address that. And of course have been very useful in turning this around. What happens if we turn the brain activation into the independent variable and look at cognitive, perceptual performance, and perhaps also introspection as the dependent variable? And along similar lines, I think neurofeedback is very attractive, and it has a different type of temporal and spatial resolution. It will allow us to probe potentially the effect of activation changes in specific brain areas, in specific frequency bands in the case of EEG, in the relationship to making brain activity similar to certain patterns of activation, and then looking at the effects on perception, on perceptual biases, for example. I have a couple of examples of that, on cognitive performance and on introspection. So I think that is a very interesting new way of using neurofeedback, not so much as a cognitive enhancement tool, which of course it could potentially be as well, but understanding of cognitive processes. So I'll talk a little bit about that. So I won't be talking as much, or I don't think at all, about the benefits of cognitive neuroscience to the improvement of neurofeedback paradigms, but I don't need to, because that's already in a very expert fashion been introduced by the previous speaker. So I'll be talking about how neurofeedback informed cognitive neurosciences, and then also, because I couldn't resist, I was giving this topic of cognitive neuroscience, but as you know from the very kind introduction that Thomas gave, my main work has primarily been on clinical applications of neurofeedback. So I will then see how we can, and have already turned these cognitive paradigms in a sort of parallel way into clinical applications. And so I hope that works. So I'll be then concluding with some recommendations also for the further development of clinical protocols, which may lead over to the following talk then. Okay, so very briefly, a general introduction into fMRI neurofeedback. I don't think neurofeedback as a principle needs a lot of introduction for this audience, so I wasn't going to dwell much on it, but those of you who come from the EEG community or MEG community, you probably know that along similar lines you can do real-time processing of fMRI signals, although of course we're talking about this different temporal resolution. We've got the hemodynamic lag, but in principle with fMRI signal we can have close to real-time, or what we call real-time data extraction and real-time feedback, for example through some sort of visual stimulus, which then is used by the participant to adjust the activation level, very similar to EEG neurofeedback. Now, what is slightly different in the fMRI neurofeedback field, it's more recent, although it's now has now had a history of 15 years and longer. Of course, it hasn't got quite the same tradition as EEG neurofeedback, and I think it's also much less standardized. So we were talking about this last night. I think that it's very rare, if it happens at all, for two labs to be using the same fMRI neurofeedback protocol. I think it's much more frequent, also because you have more commercial providers who offer standardized solutions, but also just the scientific community has developed standardized protocols much more in EEG neurofeedback than in fMRI neurofeedback. And the fact that that hasn't happened in fMRI neurofeedback as of yet is certainly, may not be bad for cognitive neuroscience, that may be perfectly fine, certainly hampers clinical translation, and that might be something that the community may want to address. So what I prepared as my next sort of slide here is a general classification, in very broad terms, of how people approach fMRI neurofeedback, and much of this would apply to EEG neurofeedback as well. So, and these are only three categories along which we can classify these paradigms, and that just shows you how difficult it is to agree. You will all have preferred, if you work in fMRI neurofeedback, you will all have preferred approaches here. So what do we target? The initial work started in what may be the most obvious thing to do is take the average activation of a region, or perhaps the differences between two. Nick Weisskopf's early work looked, if I remember correctly, at the difference between the cingulate and parahippocampal place area, but certainly these types of targets, and many people have used difference between parahippocampal place area and fusiform face area, for example, or just as average activation in a single area, up or down regulate. Okay. Many people think that that for many applications will not be sufficient. One needs to look at network metrics. So one would look, number two, at some measurement of some measure, some parameter reflecting the relationship between two areas beyond just a simple difference. For example, partial correlation or other measures derived from models of association interaction between areas. And then moving beyond that, you could also look at multivariate patterns across larger parts of the brain, whole brain, for example, multivoxel pattern analysis and its output as the real-time signal that you then use for your feedback. And so this is, in broad terms, probably captures what people are doing. Then what are your instructions? Do you essentially use neurofeedback as brain-enhanced imagery and give fairly specific and explicit instructions, and you know, try and improve your face imagery by increasing activation of the FFA? So that is something that is done. But you could also have what I would still call explicit, in the sense that people know that they're engaged in a learning protocol and have to change a target in some way, but without extraction, so in more operant fashion, which is the model that Bierbaum's group have always promoted. Or you could make it completely implicit or covert, where people are changing their brain activation and maybe learning to do so without knowing that they're engaged in a learning neurofeedback exercise. And that has been done relatively little, but I do have an example of that. What do you use as feedback signal? Do you use a bar or some other analog or symbolic representation of the activation level, or do you use more the real life stimuli, which may make it more engaging, but also more, in therapeutic applications, more relevant, but also in terms of the cognitive bias, perceptual bias, is more relevant to your protocol. So these are all choices you have to make before starting a neurofeedback study. And as I say, that already shows you how difficult it would be for people just to come up with the same protocol without aligning themselves beforehand. I'll give you some examples of paradigms that have been used in cognitive neuroscience, always sort of looking at how they fit into this model, and then similar approaches in clinical therapeutic application, without having the aim of giving you a full survey of the cognitive neurofeedback field, which is already too far advanced and expanded to be able to do that, let alone the clinical field, which is even bigger. But you'll hear about certainly the latter in, I think, the next section of this morning session. Okay, so let's start with my first example from cognitive perceptual neuroscience. A recent paper that applied what I would call here the approach probably 1 in my classification, 1, capital A, small a, and I'll just going back. So this would be a study looking at average activation, in this case difference between two areas, so category 1. They used explicit instructions. People knew they were engaged in a face scene paradigm, and they had just a sort of abstract feedback. So what this study did, which came out, I think, in the recent special issue in Neuroimage, volitional modulation of higher order visual cortex alters human perception. So they had 10, I think, participants, and they divided them up in a group that was instructed to train upregulation of the FFA from face area over PPA, or vice versa. And then they looked at one of their outcome measures was in a binocular rivalry paradigm, the dominance of faces over houses. So they had binocular rivalry, superimposed face sort of house, face scene stimuli, and then they looked at, you can't read it very well, but so on the left-hand side, you see the target areas, the FFA more lateral on the lower aspect of the temporal lobe, the PPA more medially. So one group learned trained upregulation of FFA over PPA, the other group the other way around. And then they had, this shows you that this differential feedback collapsed across the two groups. So the preferred category in the green line there, you see that over time people managed to train this differential signal. So they were able to differentiate FFA versus PPA signal in the desired direction. And this shows you the perceptual outcome measure. If you look at so you can have 3 classifications in your binocular rivalry task. You have the face percept, you have the house percept, or a mixed percept, and they pooled this again in the trained versus non-trained category. So you see the blue and red bar relatively even, so there was relatively balanced face or preferred, non-preferred stimuli, so no difference. And then if you look at post post-training, you see that if the red category went down. So the strategy unrelated percept, so if you trained the upregulation of FFA, the scene, or if you trained the upregulation of the PPA, the phase went down. They didn't, I think they'd also expected to see the blue category, blue bar going up, strategy related percept. Didn't happen, but the strategy unrelated percept went down. Instead, the mixed category went up. So a small effect of a lasting change in the perceptual biases induced by this training. So just to give you an example of how this relatively simple neurofeedback training paradigm could tell you something about the relevance of the differential activation between FFA and PPA for phase versus house perception. Now, we probably knew that already from the many studies that have mapped out these areas. So, but as a paradigm, is a sort of novel way of using this methodologically. Second example is moving to, in my sort of little classification here, to 3 in terms of the target pattern. So this would be a multivariate activation pattern, and B means a sort of operant without a predefined strategy, operant training, but again with just a sort of abstract stimulus. In this case, an increasing disk, and this summary there on the left side relates to a technique called DECNEF, which many of you will have heard about, which has been reviewed in this review paper by Watanabe in Trends in Cognitive Sciences 2 years ago, and it refers to the training paper which was published by Shibata et al., Perceptual Learning Incepted by Decoded fMRI Neurofeedback Without Stimulus Presentation. So left, you see the basic idea of this DECNEF approach, which is online decoding of a pattern. In this case, they were showing different gratings of different orientation, then they used activity from the primary and higher visual areas to distinguish between these different patterns which worked, and that in itself had been done before. But then in this particular study, they trained participants then with neurofeedback using this, training them, or their task was to change the size of this disk by enhancing activation corresponding to a particular orientation, one of 3 orientations. So that was the training paradigm. And then afterwards they showed, if you look at the discrimination performance there, there's in plot D, for example, you see that the perceptual discrimination improved most for the targets, but not for off-target stimuli. So although during the training they hadn't actually seen these gratings, they were after training for the neurofeedback training for this particular orientation, were better at discrimination just for the specific target orientation. So that was one of the first demonstrations, I think, if not the first demonstration of actual perceptual learning aided by neurofeedback, by fMRI neurofeedback. Third example, again, multivariate pattern, so it's number 3. Back to explicit cognitive task, attention task, cognitive instruction, so capital A, but now with a task-relevant stimulus, in this case, face-house stimuli. So you notice something here, a lot of, again, this is something going back to what has cognitive neuroscience or perceptual neuroscience ever done for neurofeedback. Well, this very extensive work mapping out FFA, PPA, the properties of these areas, specificities, and also multivariate patterns, if you think back to Haxby's early work in this domain, has really given us the targets to essentially start with robust activation, easy to localize, relatively easy for people to modulate with explicit imagery. So this is a good training ground for participants and researchers before you move on to perhaps less well-charted territory. So again, faces houses. This was a study here de Bettencourt et al. From Princeton, closed-loop training of attention with real-time brain imaging. And here, essentially, the face of it, people were doing an attention task. They had the superimposed stimuli of houses or scenes and faces, and they had to attend to the scenes, respond to indoor scenes, inhibit responses to outdoor scenes. So this, of course, in order to do this, you have to attend on the scene component of that superimposed stimulus. At the same time, they were tracking the scene-related activation based on a multivariate classifier that they had trained, and whenever they essentially noticed an attentional lapse in neural terms. So whenever they saw that the pattern was moving further, towards the wrong category, they were attending more to the faces. The task essentially became more difficult by the scene percept becoming sort of harder to differentiate. So. Hang on. I don't want to. No, I think in this case actually there were. In this case actually there were, if I remember correctly, this one, because both you could do both. In this case, where actually I think they were rewarded by whenever they had the sort of right attentional pattern, the stimulus became more scene-like. I think that's what's reported here, and with this neurofeedback enhancement, so you got the change in the stimulus properties based on the neural activation pattern. They actually showed an increase, a small one but significant, in behavioral performance A prime in the feedback group, but not in a control group. So this was a way, I think one of the first ways, again, of using essentially classical attention training, but enhanced by neurofeedback, in the sense that in a closed loop fashion, the target stimulus changed dependent on your brain signal. And apologies, I can't give you the full, don't remember the full details just now, but of course you can look it up. But I think the take-home message here is you could potentially do it either way. I mean, if you think that you want to sort of perhaps sort of shape the training by giving people, making the training harder in small steps, you could do, you could make the stimulus harder to discriminate whenever people move up to the next neural level, or you could also reward them by shaping the activity in the right direction, which I think is what they did here, and that if you go into the right attentional pattern, you make the stimulus easier to discriminate. So based on your learning model, you could move it in both directions, which I think is potentially very attractive in terms of combining or enhancing attention training or other cognitive training with a BCI-based change in your stimulus or in your it doesn't have to be a change in stimulus. You could also make your stimulus presentation faster or slower based on the neural state. So I think there's very interesting manipulations. You could do very interesting combinations of a neural state and your stimulus parameters. My last example from the classical cognitive neuroscience is a covert neurofeedback, or sort of implicit neurofeedback. And these terms are used in different ways. The DECNIF community also call what they do implicit, because people don't have a strategy, but mostly they still know that they're engaged in a training protocol. So it's a bit of semantics. But what I mean here by implicit is what was called covert by the authors. Covert neurofeedback without awareness shapes cortical network spontaneous connectivity. This study did not have cognitive outcome markers, but they looked at neural effects. But what essentially the novelty of this design here was that participants were in the scanner. Again, this was a univariate feedback difference, again, between FFA and PPA. You see a theme here that is just very nice to use these areas, and again, the difference, which you could manipulate by differential imagery of faces and places, for example, if you knew what's happening. These participants didn't know where the target feedback came from. In fact, they didn't even know that they were engaged in a neurofeedback paradigm. They just knew that at certain intervals heard rewarding sounds, and they were informed that this was a study of reward. But in fact, most of them, I think 10 out of 16, okay, well, actually increase the frequency of these rewarding sounds by changing their FFA PPA pattern in the desired way. But even those who had done that were not aware that they had actually done this, nor were they sort of reporting an explicit strategy. So they didn't seem to be engaging in an overt face or place imagery paradigm. And so that was sort of one of the first studies looking at this, and they did extensive mapping studies of functional connectivity changes, which I'm not bringing in here because they're not part of the sort of central message as such that you can do this. So although not everyone will be able to do this, with only a proportion of their participants, and the activation change here that or the concurrent activation that's plotted on the right side is quite interesting, where you get auditory cortex. In fact, this is a difference between the rewarding and non-rewarding stimuli. They interpret that as perhaps reflecting the increased salience, but also you get what we would commonly interpret as reward-related areas here in the stratum. So that would, although that pattern probably would not be different necessarily if you just were studying reward, it may not be directly related to the neurofeedback component as such. But this is certainly very interesting. They had a go at this, and we're also looking at this at the moment. My colleague Leon Skotnik has recently worked on this with Rainer Goebel as well, on differentiating the learning-related signals in fMRI neurofeedback from those responding to a reward. Now, you would expect when you're informed about a reward, particularly an unexpected one, to have all the prediction error processing areas, ventral stratum, activated. But if you differentiate in time preceding this, you should have the learning-related activation in a similar network before you are informed of the reward. I haven't got slides on this, but I think that's that's an interesting direction, again, specific to neurofeedback, because you are the generator of the reward yourself by your neural activation, which is quite unique in terms of the paradigms that you can do. Okay, so these 4 examples gave you, I think, an overview, without claiming to be comprehensive here, of the various manipulations that you can do, some of which are similar to what you can do with EEG, some are specific to fMRI. I think in terms of the targeting of certain networks that you can do, combining them with cognitive paradigms to really study cognitive neuroscience, not just in the sort of enhancement idea, but also in a mechanistic way. And I think this is, as you also see from the prominence of these papers, this is actually an interesting and growing field. But how do we move then with these paradigms to clinical translation? And I'm not actually following the chronology of the field, because many of the clinical applications had actually preceded the cognitive applications, but they are governed by the same principles, often same areas, and this sort of general classification of whether you use a univariate feedback, functional connectivity, multivariate or a signal, and so on. So I suppose the simplest, in a way, although not necessarily simple in its implementation, but perhaps conceptually, would be 1AA, which is you use a univariate signal, you give explicit instructions, and you use a symbolic abstract feedback cue. This is what we and also other groups have done in relation to developing paradigms for depression. So here what we've done is use a localizer based on IAPS pictures, International Affective Picture System. So you single out the areas or a network, summary activation of a network responsive to positive images. And then during neurofeedback, you instruct people to increase activation in these areas. And you do this with explicit instructions to remember the localizer stimuli, start from there, engage in positive imagery. So this is now neurally enforced positive imagery with desired cognitive element, not purely operant. And although you could also then use these positive scenes as a stimulus that would create visual confidence, so we've kept it simple and just used a thermometer upregulation target. So this was what was done in one group, NFE, neurofeedback emotion. This is data from a recently published clinical trial. And you see here the target area, which varied from participant to participant, session to session in limbic and paralimbic areas, insula, for example. And the control group, again, back, you will recognize this by now. Number 2 is the parahippocampal place area. So a control group did another veridical neurofeedback paradigm, not sham, but an actual neurofeedback training paradigm targeting the parahippocampal place area. And again, they knew that this was identified based on scene stimuli, and that scene imagery could be useful strategy for this. And actually, both groups managed to do this, and they managed to do this equally well. That's, of course, a challenge to come up with a control condition that people can actually achieve and do equally well, so you don't end up with differences in performance, reward, self-regulation experiences, which could be confounds. So we think this was a reasonably well-controlled study of, actually, I think a very well-controlled study, which also appears from the clinical data, because although there was substantial improvement in depression scores pre-post, there was no difference between these groups. And now there, as if you look at the left plot here, Hamilton depression rating scale went down from around 20 to around 10, and that was sustained after follow-up, but no group difference between the NFE and NFS group. Now that sort of two basic interpretation of this, either without, because we didn't have any other sham group, we didn't have a treatment as usual group here. So both of these neurofeedback protocols may just have worked remarkably well, or what we're seeing here may be non-specific effects combined with spontaneous remission. We have a slight bias to favor the first interpretation, but then we're, as investigators, most susceptible to biases, as you heard before, or as but based on the size of the effect, I think this is probably more than what we would call a placebo effect. But of course, methodologically, this would have to be proven. This is a big debate in the field at the moment, just based on what kind of control condition you can show this, and we'll come back to this later. Another example, so this was what probably most people up to now have done, use a single area, relatively sort of abstract stimulus as feedback. We've done it in depression, we've also done it in Parkinson's, and other groups are using these types of paradigms as well. But as I said, unfortunately, and there still needs to be standardization. I'll come back to that later. Okay, so this is now another approach which is coming out of the DECNEF community. So you have a multivariate, 3BA, you have a multivariate signal, B, it's an operant paradigm, and in this case, again, the stimulus was the sort of increasing disk, so sort of abstract feedback. So this was a paper that was sort of intended to be, I think, on the pathway to developing alternatives to current treatments for anxieties, or specifically for phobias. So that classifier was trained on different categories of animals. including the one that people had a specific phobia to. One methodological twist here was that they used what's called hyper-alignment, so they used a classifier that works across subjects, which circumvents the need to construct an individual classifier, which can be elegant, particularly in anxiety disorder. So if you wanted to identify in individuals' brain pattern associated with, say, a snake, if they have a snake phobia, generally what you would do, you'd show them different images of snakes, and then other animals that perhaps other reptiles they don't have a phobia for, and then you train the classifier to differentiate these two. That's the most direct way of doing this. Of course, the problem there is that you have to expose the patients to quite a few images of snakes, and they may not like that. And they may react aversively, or even if they like it, you may get anomalies in your response. They may move more and so on. So this hyper-alignment idea, training the snake pattern on other people and transferring it, may be attractive, but of course you lose some of the anatomical specificity. They did address that in their method. So let's assume for now that that's fine. So they developed with the online decoding essentially a target activation pattern that would respond to the feared animal category for these participants, and then used it for a group of patients with a specific phobia, who were then trained actually, or their target was to, again, increase the disk based on the activation pattern responding to their feared category. So their task was actually to, they got rewarded with money for whenever the disk became larger, and that was contingent upon the occurrence of this fear-related brain pattern. So the idea would be you do an exposure therapy without actually exposing them to the actual stimulus. So it's implicit in that sense that they are exposed to the brain pattern without actually seeing the snake, which may make it less aversive, more acceptable to them. So I think that's a very elegant idea. And they did show then, after this training, in their arousal measures, both in terms of the bold response in the amygdala and the skin conductance to these feared animals, reduction in the arousal measures for their specific animal category in the trained group, but not in a control group. So in this sense, this neural exposure training was successful, at least using these surrogate markers. It was not a full clinical trial. They didn't go that far into sort of clinical outcome measures. A slight caution, I think, if you dig into the supplementary material, you'll see that the training effects were very small, if present. There was a difference between the two categories, so target and control, but it hovered around the initial where it was before the training, and also there was no significant interaction between time and condition. So as you would expect, it's actually hard for people to train these kinds of patterns in the absence of the original stimuli. So I think that this is always a slight limitation for these MVA training approaches, that although perhaps significant, the training successes are often, the classifier discrimination is often quite small, and then corresponding the training success as well. But it's a very, very attractive paradigm, I think, especially to think about anxiety disorders. My third example in terms of the different paradigms people use, again, goes back to relatively simple neural measures, univariate average activation, relatively explicit cognitive instructions, in this case around motivational stimuli, so cognitive reappraisal of a target, which is also on screen. So that's the novelty here. You actually do the neurofeedback while you look at a stimulus. We piloted this with food and drink stimuli originally, but you could equally do it again with fear-related content, with emotion-related content, and then make the salient stimulus change contingent upon the brain activation, and for example, make it disappear, make it become smaller. This is something that we did in a pilot study in relation to craving in healthy individuals who had fasted for half a day, and then during lunchtime where in the scanner, so we mapped out the salience respond to appetizing food in their striatum, amygdala, limbic, paralimbic areas. I'll show you some examples, individually localized, and then they were instructed to downregulate the activation in these areas and then see the food stimuli become smaller. So this would be a paradigm, again motivated by the sort of cognitive approaches of approach avoidance that are being used in motivational disorders. So this could ultimately potentially be a paradigm that you could use, you know, for the treatment of obesity, although we're not there yet. This was absolutely a pilot study in healthy individuals. But what you see here in the 10 people, it was individually localized amygdala and many other areas, basic ganglia, so on. So the individual target area was used, and then they managed to downregulate it. We compare because, of course, you can have decreasing activation just by repetition of stimuli. So we had a control condition where they just passively viewed the stimuli, and compared to this, they actually managed to downregulate the activation in these target areas, which was also accompanied by a slight reduction in sort of craving scale, but not in objective food intake. So I think this was just mainly sort of to pilot the paradigm. Whether this kind of intervention can ultimately have an effect on sort of attenuating motivational behavior, we'll have to see. We are in the process just at the moment of analyzing data from a trial where we use this in alcohol and in abstinent patients with alcohol dependence. But I think in principle it's an attractive for these sort of cue reactivity disorders, but also for stimulus-dependent anxiety disorders. This could be an attractive paradigm. Okay, my interim summary now. Just to get some order into this field of fMRI neurofeedback, I gave you my sort of taxonomy according to target, strategy, feedback signal. And in this first category, quite a lot of work has been done, particularly in depression, our own work, and also work trials done by Kimberly Young in the US. I told you a bit about the DECNEF, which starts from the multivariate recognition of patterns, for example, related to particular fears. I've told you about the motivational neurofeedback that, for example, we've used in eating and relation to alcohol use disorders. And what I didn't mention but should mention here is that quite a few people are also now using functional connectivity, mainly with operant paradigms, mainly with abstract feedback, so frontolimbic connectivity, for example, for clinical applications. So just to mention a few current challenges in the last few minutes of my presentation. As I said, the protocol development so far has been idiosyncratic. Most groups, again, that's probably because of the cognitive neuroscience roots of this field. Similar to cognitive neuroscience, everyone has their own topic that they study, and they develop their neurofeedback paradigm around this. The development was not driven by clinical standardization, and that's fine if you want to do cognitive neuroscience, but a limitation if you want to apply it to the clinic. For evaluation, picking the right control condition is a huge challenge. And then to the next, if you want to go to the next step after going through these initial, relatively small studies, a huge challenge for the field to move to adequately powered efficacy studies. And I guess we'll discuss that a bit later when we've heard about the primarily clinical presentation. In terms of standardizing protocols, there's, in collaboration with Talma Hendler's group, there's been a recent paper in Nature Human Behavior which proposes a process-based framework for neurofeedback, which is quite interesting, which is motivated by the RDoC classification. So again, apologies for the slides, probably too small for you pass. The basic idea is that rather than developing individual paradigms for individual disorders, you develop a paradigm that can actually target several of the RDoC domains. Like in this case, the yellow is cognitive control, there's approach motivation, there's fear avoidance, which can all be related to depression, dependence, and anxiety. And then whilst you navigate in the middle here, whilst you navigate a immersive virtual environment, you can do various things with your brain activation. So if you increase your striatal activation of the striatum, an appetitive stimulus will become larger, which may be desirable in terms of depression, or if you downregulate your amygdala, an aversive stimulus may become smaller. So you could sort of incorporate in one paradigm different of these RDoC domains, and then use all of that or individual targets depending on your patient's needs. The choice of control condition is a huge challenge. There's been, again, the special issue now, a paper, and also in this paper, discussion of what to do. I still think that using a different target area is a relatively good and conservative feedback. Here in this paper was proposed not just to focus on one target area, but use a randomized set of target areas for the control group, which in principle is a good idea, but their criteria are control areas that are equally easy to regulate that as your target area and non-relevant to the disease process. Now, I find it hard even to find one of those. It's difficult to find more than one. The PPA certainly was equally easy, but probably not unrelated to depression. And after we'd started our study, a study from the National Trust, a big owner, sort of charitable landowner in the UK, came out that being in the open environment and imagining scenes is good for your mental health. Now, of course, had we known this beforehand, we may not have picked the PPA as a target area. So it's very difficult to think about the control. And of course, there's a big push, and I can see absolutely how this is coming to have sort of almost molecular level controls. Control for each individual process, make sure the non-specific effects of machinery, the self-regulation experience, the reward experience, the mental imagery are all controlled for in a neurofeedback paradigm. Now that, if you really want to fractionate these processes, that makes sense. But it's very hard to do, and you could end up with a 5-arm trial, which is completely impossible to run. You could also, if you move into sham control conditions, move into scenarios where actually you don't know whether your control condition is actually harming the patient, or what it's doing, or hindering their improvement. So you could have artifacts from your control conditions, and control conditions in complex interventions and involving psychological processes are rarely inert. So I think controls are important, but if I just sort of can give you one example. No one, if we talk about a pharmacological study into an antidepressant, say a tricyclic antidepressant, I don't think anyone would demand that you do control, that you run control conditions that will tell you whether the serotonergic or the anticholinergic effects or the effects on the histamine system of an antidepressant give you the therapeutic effects. No one would tell you design specific agents targeting so you have a tricyclic that may have affinity for 5 or more receptors. No one will ask you to design individual drugs targeting just these receptors to really know which receptor interaction your therapeutic effect is based upon, which is, however, something that we're being asked in neurofeedback. So that's why my philosophy at the moment is to first start by showing that what we do in neurofeedback is superior to treatment as usual, which is, of course, a clinically relevant question, and then start sort of breaking it down in the actual mechanism. But that conflicts with the interests of cognitive neuroscience, where, of course, we would like to know the specific molecular features often. Yeah, just to sum up, these are slides I'd prefer more for sort of in a clinical context. Perhaps I can just show you the most salient one, which is the challenge of numbers in terms of the effect size, particularly in depression, that we're looking at generally when we do a placebo comparison. We're talking about effect sizes in the range of perhaps 0.4, 0.3, which immediately gets you into, with conventional hypothesis testing designs, into trials of several hundred participants. This slide prepared by David Mehler, who came to one of your days here a few years ago, completed his PhD in the meantime. He's very interested in using Bayesian sampling to improve or to make it more practical to run these trials. But in any event, moving, and particularly with fMRI, these trials can soon get expensive. So from a clinical perspective now would be our general recommendations for the field. Some of these are fairly generic. Pre-registration we do in trials anyway, and we might want to do this more for the earlier stage work as well. So we're now doing this for our pilot work as well, a small trial, small pre-trial study in stroke, for example. We've pre-registered. So I think that's where the field is moving anyway. Standardization, I think that's still something where certainly in fMRI we're lagging behind the EEG field, both in terms of paradigms, outcome measures. Now for outcome measures that may be easier to do within the Braintrain consortium, we've agreed on cross-cutting outcome measures, also predictive measures, where we could now then start addressing the question of the literacy or illiteracy for neurofeedback with personality and other performance measures. So I think that is something where the fMRI field can learn from the EEG field, but also standardizing outcome measures. Standardizing protocols will be more challenging. We've started something, an exercise in the fMRI neurofeedback field with the template for intervention, description, and replication a couple of years ago, and we might repeat this again just to map out what people do before we can move to standardization. We need to know the practices of the field. So I think this is something where also conventions like this, I think, can be very useful. So let me just conclude by thanking you for your attention, inviting you all actually to the next real-time fMRI and neurofeedback conference in Maastricht and Aachen in December. There's a Twitter feed that you can look at. The registration is open. Bettina Sorger is the program chair. She's very interested in workshop proposals. So it's not very far, actually. It's only six and a half hours by train from here, even closer from Paris. So you might consider coming along. Our Braintrain consortium certainly had a very good time at the NARA meeting. Some of you were there. This is also recognition that a lot of the work that I showed you grew out of the Braintrain consortium collaboration. Thanks very much.